All right, chemists, let's get started. So a couple more days till the end of the term. So the outlook here, we will finish chapter 10 today. There's just a couple slides left. The homework is due on Monday. I moved it because I don't know if you noticed, but there was like a huge Pearson outage yesterday. It was broken. It was broken for everyone though, okay? It was even broken for me because I was trying to put together the homework for nucleic acids, which is up now. So that's chapter 11. And I decided that that's gonna be our last chapter, all right? Because I think it wraps up a lot of the concepts and ideas that we've been talking about thus far. We'll start chapter 11 today, and I'm gonna throw out some of the sections of it, and I'll make it really clear what is expected of you and what's not. Basically, if it's on the slides, then it's free game for the exam, for the final exam, all right? So that's our last chapter. We're starting it today. The homework's already posted, and it's due on the very last day of classes, so Monday. It's going to be due on Monday. And I'll make more announcements later, but for any late homework assignments, so if you have like one or two questions you haven't done in previous homework sets, remember you can still complete those questions for partial credit. It's not a lot. The maximum you can earn is 10% of the original grade, but that's even if it's like 3 months overdue. You can still complete those. The last day for late homeworks is Tuesday, which is reading day, at 8 a.m. So again, you can, grab those, any extra points that you can for the end of the term. Okay, so chapter 11 is our last chapter. And then finally, the Zoom final review that I'll have, the 2-hour one that I do, I need to write up some document that we work through, will be Tuesday at 6 p.m. So Tuesday's reading day. Do you know what that is? It's basically the last day of classes is Monday, and finals start on Wednesday. So Tuesday is like the last deep breath, before finals start. And so there should be no finals or anything on reading day on Tuesday. So I'll have that review session on Tuesday because your final is on Thursday. So that gives you some time. I will post the document, the key, and the video. All right? Sound good? All right, so let's get started. We left off talking about proteins, and a lot of the same themes are coming up in this proteins chapter and in the next nucleic acids chapter, where we're taking these smaller building blocks and building up larger strands of things. So in this case, we're building up amino acids, the small building blocks, and building these longer strands of peptides, polypeptides, or what we call proteins. We've said that proteins have a lot of different functions in our bodies. Proteins are enzymes within our bodies. They have lots of functions. They can do things structurally within our bodies. They can store things in our bodies. Also, a lot of hormones are actually just big proteins. So building proteins, super important. So this slide where we talked about enzymatic hydrolysis, this process is breaking down those large protein strands into smaller building blocks. So it's like Legos, right? Like you're taking a complete set of Legos and breaking it down into smaller pieces so you can go build something else. That's like the whole process of like eating and metabolism. We eat proteins so we can break down to the single Lego pieces, the building blocks, to build up things that are useful for our bodies and the functions that we need to survive as human beings. So we talked about let me just go back a slide. How we can break down proteins. So recall lysis is the opposite of condensation. Condensation, we bring things together, remove water. Hydrolysis, we add water to split things up, to break it. And so we talked about hydrolysis of larger molecules in chapter 4, where you just heat the thing, you add acid, and it breaks apart. But within the body, how hydrolysis can occur is actually through enzymes, specially designed enzymes that can break down specific amino acids. So we talked about 3 different ones. And the thing to note here is that all enzymes end in the suffix ace. So that means enzyme, that it does some type of function there. So we have 3 different ones: aminopeptidase, carboxypeptidase, or endopeptidase. And that just tells us what part of the protein strand does that enzyme cleave off to make amino acids. So aminopeptidase cuts off the N terminus, carboxypeptidase, the C terminus, and endo is anywhere in the middle. And so we looked at these molecules, these longer peptide strands. And so what aminopeptidase does is it cleaves this peptide bond. So there's kind of 2 ideas here to note. It's, can you identify where the peptide bond is? And I told you before that what you want to look for is a carbon, a carbon that's attached, a double bond to an oxygen. So look for that. And where that carbon, that carbonyl carbon, is attached to a nitrogen. So that bond that connects that carbonyl carbon and the nitrogen is your peptide bond. So all of these these enzymes break those different types of peptide bonds. So this one, because it's on the N terminus side, will be cleaved by aminopeptidase. This one will get cleaved off by carboxypeptidase, and all these middle ones will be done by endopeptidase. All right, so there we go. All right, other ways that we can break down proteins, again, to the whole role here and the whole purpose of breaking down proteins is to get smaller building blocks so we can make something else more useful in our bodies. So these enzymes, these protease enzymes, basically the same thing as the enzymes that conduct hydrolysis. The whole idea here with these enzymes is that they're just protein specific. They still break down proteins via hydrolysis into smaller units. We just give them different names because they have specific function. They work on very specific proteins. So these proteases also are enzymes that do hydrolysis. It's just like a fancier name for them. So some common ones that we have in our body: pepsin. This enzyme is in our stomachs and will break down animal plant proteins. All right, so it's just a little bit more of a generic term than the other 3 enzymes we talked about that cleave specific spots on an amino or on a peptide chain. These ones just kind of work more generically. So pepsin's in our stomachs. It breaks down meat, eggs, seeds, all that stuff. And then renin is another type. That's the common name of this protease. It breaks down, it's an enzyme that's found in the stomach of young mammals, and it's actually the enzyme that's responsible for making cheese. Has anyone ever tried to make cheese before? Like from milk? No one? Okay. But so what you do is you add a little bit, so you take like raw milk, like it can't be homogeneous. You can't use the milk that's at the grocery store. Like you can't go to Kroger and just use the milk there. You got to get like milk, like fresh from the cow, all right, raw milk. And then you add a little bit of an enzyme, and what happens is that the proteins within milk curdle, and you collect all of that, you know, like the curds and whey. You all look gross, but this is how cheese is made, all right? You separate the curds, all right, like the chunky parts, okay? But that's the tasty part, okay? And then you separate it from the whey, but the reason that separation happens is because of enzymes. And this enzyme is in the 4th stomach of mammals, so like in the intestinal parts of cows. So that's how cheese is made, all right? And so the enzyme, you put it in there, the curds separate from the whey. On an industrial scale, they have like huge vats of this, and they strain all that whey out and collect the curds, and then they either let it age or dehydrate a little bit until it gets the cheeses that you want. Fun fact, I just thought of this. What state is the cheese, when I say like the cheese state? Wisconsin, yeah, people think of Wisconsin, all right? And so recall I'm a chemist, and I like to tell this story. So in the winter time, what do we put on the roads to prevent it from freezing? Salt, yes. Some people say ice. No, it's salt. Salt, sodium chloride. So what we're doing is we're putting something that's salty on the roads, okay? So when you think of cheese, you think of salty stuff too, right? Like cheese is salty, yeah? Okay, so the whey The water product that comes from cheese after you add the enzyme, all right, is also very salty. All right, when you take away the curds is the stuff we eat. The whey is the stuff that a normal person would just throw the whey away because it's just water and no one wants it. But in Wisconsin, because they make so much cheese, they spray their cheese byproduct whey on the roads in order to not have them ice over. Yeah, so their roads are a little bit cheesy, all right? So instead of just throwing it away, they put it on the roads, okay? I'm sure they put salt as well, but it's just kind of like a reuse, reduce, recycle type thing. Yeah, if you throw it away instead, okay. All right, so there's my side story. All right, so there's some more functions of protein. So these proteases also just break down larger proteins to smaller ones that we can use for other purposes in our body. All right, so another type of protein is hemoglobin. All right, so this is the protein that's responsible for moving oxygen within our bodies. And so hemoglobin is a good example of showing the quaternary structure of proteins, because in order for hemoglobin to work, we need actually 4 different protein subunits. So 4 different strands of proteins to come together to work together in a single whole unit to work as a oxygen transport vehicle. So what you see here in hemoglobin is that there's 4 different subunits, so you can kind of see the colors here. All right, so we have 4 different strands, and typically we call these, they're 4 different helical secondary structures. 2 of them are alpha in stereochemistry, or in secondary structure, like they twist one direction, and the other 2 are beta in the other direction. So the secondary structure matters of these single subunits. But then when all 4 come together, then we make a fully functional hemoglobin protein that can do stuff with oxygen. When all 4 of these units together, they're held together by specific intermolecular forces that keep them together. But what's special about hemoglobin is that I said there's 4 proteins that come together, all right? But what else are in these proteins? So you see there's 2 reds, 2 blues, those are the proteins. What else is very special in these are these heme groups, all right? So these heme groups are iron, all right, iron 2+ and iron 3+, depending if oxygen is bound or not. So each of these iron pieces within the hemoglobin, that is the spot where oxygen can come in and bind. Those heme groups, those irons, need to be there in order for the whole protein as a whole to function. So all 8 of those pieces need to come together in our bodies to just make one functional hemoglobin. So that's the whole idea of quaternary structure, that all of these things need to come together. They need to work together as a team in order to do the things in our body that they need to do. So if there's one piece missing, if there was just 3 hemes, if there's only 3 irons, this whole thing would fall apart and it would not work. So what's cool about these heme groups, the irons, so since there's 4 iron centers here, one hemoglobin molecule can actually hold 4 oxygen molecules. All right, so each one can hold 4 and then transport it in our body where it goes. And so when oxygen binds to those heme sites, what happens with the quaternary structure is it changes slightly. All right, it carries the oxygen, it goes where it needs to go, and then the oxygens cleave off to wherever it needs to go, and then the structure, the whole quaternary structure, changes back. So there's a little bit of structural changing whether oxygen is bound or not. We could go into a ton of detail about that, but that's all you need to know, essentially. All right, so there we go, hemoglobin. And of course, the structure tells us a lot about, or that structure dictates a lot about the function of what hemoglobin can do. All right, so let's keep going. All right, so on that note, so I mentioned that iron, that heme group in hemoglobin, is iron a protein? Don't think about it too hard. No, iron's a metal, all right? But it's part of the protein. All right, and so this idea here that sometimes there's other components to proteins in order to make them function. So enzymes are a type of protein, all right? Enzymes are just a specific type of protein that has a specific function. And so we're gonna kind of switch gears and talk about enzymes and what they can do in our bodies. When there's a component in a protein or an enzyme that's not a protein, so like the heme group, we call that a cofactor. It's like a helper. To help that enzyme do what it needs to do, though it's not technically a protein. But if it weren't present, then the protein or the enzyme won't function as it's supposed to do. So we're going to talk about enzymes here. So enzymes are a type of catalyst, a catalyst that function in our body. So a lot of the proteins and enzymes like the proteases, the enzymes that hydrolyze, those are all enzymes, and they do specific functions like cleave amino acids. So they do something specific. So I kind of just glossed over that, you know. I say, oh yeah, amino acids can break apart a polypeptide chain, but like how does that happen? It's through the use of enzymes. Enzymes are like the workers who actually do it. It's easier to just say it, you know. It's like, yeah, two glycines break apart, but who did it? An enzyme did it. All right, and so we'll talk about enzymes now. So enzymes, the thing that catalyzes or makes these reactions go faster. So if we had like two glycines connected to one another, if we don't give it an enzyme to like help break that bond, it may never break in our lifetime. So what we do is we have enzymes that help facilitate these reactions that we want to happen in our body. So yeah, enzymes, body's catalyst. So the same idea of when we talked about catalysts in Le Chatelier's principle. So catalysts just speed up reactions, and the reasons they speed up reactions is because they all lower the activation energy, the activation barrier for those reactions to go forward. We'll go into more detail there. All right, so we have some cartoon models of what enzymes are. So like right here, you kind of see this cartoon model of an enzyme, but what this little cartoon is, it's a bunch of proteins. So remember that, it's a bunch of amino acids, all, you know, have secondary, tertiary structure folded in a certain way to interact with the substrates or the reactants around it. So do keep in mind that, you know, these cartoons actually represent amino acid, you know, strands. Okay, so enzymes, catalyst. So we oftentimes talk about substrates in relation to enzymes. That's just a fancy way of saying a reactant. All right, so this is just a chemical reaction where we have reactants going to products, but we just call the reactants substrates when talking about enzymes. So right here, we have glucose and ATP. We want to put a phosphate group on this glucose. These two things are our reactants, but within enzyme chemistry, we're gonna call this the substrate. These are the substrates. So there's two of them here, all right? So what an enzyme does, so when we were talking about chapter 4 stuff, and we had chemical reactions, right? We have like, we have reactants and we have products. Sometimes above the reaction arrow, we put things like, oh, you have to heat it, or you have to add acid, or sometimes we write, you have to add a catalyst. And we just, you know, very kind of flippantly just put catalyst over the arrow, but we don't think about what that is. So now we're going to look at the detail of what it means to add a catalyst. So right here, this substance is the catalyst. You know, or it's the enzyme, because we know because it ends in ACE. So this is our enzyme here. All right, so we're going to look into details of what it means to add that enzyme. But overall, what's happening is that enzyme is assisting in this chemical process where we're taking this phosphate group and we're putting it on the glucose molecule here. All right, and so on the enzyme, and this is the detail, what that enzyme does is there's this pocket within that enzyme, because remember, it's a bunch of protein strands that have certain folding and tertiary structure. There's a pocket within that enzyme where the substrates can come in and interact and then have the chemical reaction go forward. So the pocket within the enzyme is very, very important here to facilitate that reaction. All right, so there we go. So right here, that's right here on our enzyme is our active site right there, where the substrate or the reactants fit into the pocket. all right, and it has, and the enzyme has a pocket, has an active site because of the folds, an idea of tertiary structure from the proteins that we talked about. All right, keep going here. So substrate specificity. So what that means is, okay, does the enzyme recognize only one thing? Or does it recognize like a lot of different sugars? So for example, this enzyme, we see the reaction occurring with glucose, but if this enzyme also works with, let's say, another sugar like fructose or mannose or something, then we would say that there's a low substrate specificity, that the reactant just has to look like not something specific, but just look like a certain type of shape or whatever, that the enzyme isn't, I guess, I can't think of the word. Doesn't have like a preferred one. Don't all look at me at once. The enzyme can recognize a lot of different substrates and is willing to work with any of them versus just, okay, no, I need glucose. No, actually, I just will work with D-glucose and that's it, and I won't recognize anything else. So that's very high substrate specificity, right? Versus, no, I'll just do any type of sugar, it's fine, you know? All right. So, and then right here, cofactors and coenzymes. So these are the ideas of helpers. So cofactor is just anything that helps with this enzyme catalysis that is, we typically say inorganic. All right, so for example, the heme group in hemoglobin would be a cofactor. Because the iron is present. So it's just a helper. It's not a part of the enzyme, but it facilitates that chemical reaction. So that's a cofactor. A coenzyme is the same idea, that we have a helper that's not a protein, but a coenzyme is not inorganic, but it's just a small organic molecule. So the reason, your doctors say like to eat your minerals and vitamins, all right, so minerals implies inorganic, like small, like group 1, 2 metals that we ingest. Those are kind of like the minerals, the cofactors required for enzymes to work in our bodies. Vitamins are larger covalent molecules. All right, like if you look at the structure for vitamin A or vitamin B, they're larger covalent molecules, all like carbons, hydrogens, and oxygens, and so forth. Those are coenzymes. So they both facilitate this enzymatic catalysis. It's just cofactors have like inorganic, are made of inorganic substances, so metals kind of on the periodic table, versus coenzymes, also helpers, but they are made up of non-metals, so they're molecules. So yeah, this is the idea of like vitamins, eat your vitamins and minerals that you need to survive, all right? Okay, and they help with the larger proteins, the enzymes, to do the catalysis that we want. Okay, so keep going. All right, so the details of this arrow, like it's so easy to just write, okay, yeah, at a catalyst, this reaction goes. So we're gonna focus on that arrow. What does that mean? So here's a picture of what is going on. So do understand this notation that I have here. It's another way of saying this reaction that I have up right here. So E means enzyme. S means substrate. So what this reaction is saying is, okay, you have a solution, which is our bodies. You have enzymes and substrates in your body, all right? And they're not attached to one another. But eventually, because of chemical equilibrium, the enzyme and the substrate connect with one another. And so what the double arrow is showing is like, hey, that's an equilibrium. And what we produce is this ES complex, where your enzyme and your substrate are attached to one another. So they're locked into one another. And so that is kind of what this picture is showing right here. So this really should be a double arrow. That in this picture, the glucose is not in the pocket of the enzyme yet, so they're separated. So that's why it's E plus S, they're separated. But then once that glucose goes into the pocket, now we have this enzyme substrate complex. All right, this new substance. And so this is where we call it the ES complex. So now that substrate, that reactant is stuck in the pocket. So now that it's in the pocket, the enzyme can go and do its work to facilitate the chemical reaction that goes forward. And so that's what you see here, this chemical reaction arrow, that's the full arrow. Once we have this complex, the enzyme substrate complex, the chemical reaction can occur. And then what happens is that we create the product, and then it can go away, and then the enzyme can do it again. All right, so that's this whole idea. So this complex is not really implied when we write chemical reactions like this. It's not really implied, but it is happening that way, that when we write an enzyme above a reaction arrow, this is what we mean is happening, that enzyme is helping bring together these two substrates so we can facilitate that chemical reaction going forward by making this enzyme substrate complex within there. So that's how we read that. All right, so the different models of how enzyme catalysis works. So again, the cartoon model here, we talk about the active site within the enzyme where the substrate or substrates go as a pocket. So there's different models to kind of explain enzyme activity, and so one of them is the lock and key model for enzymes, where it's just as simple as that we have a pocket, it has a certain shape, and if we have a substrate that comes in that fits that exact shape, it will go in. But the idea of the lock and key model is that pocket is rigid, so only one type of shape will fit. Does that make sense? Versus a, what's the other one? Induced fit model, all right, where again, thinking about the active site, it has a shape, but it's rigid, or sorry, the lock and key is rigid. The induced fit is a more flexible active site. So that idea is, okay, we have this really flexible active site, so if we have a substrate that comes by that kind of fits in the pocket, it'll go in the pocket. So with that idea here, we have a rigid active site, that's lock and key. Then we have induced fit, that's a more flexible active site. In which of those do you think, which model do you think explains an enzyme with low substrate specificity? So if a substance has low substrate specificity, it doesn't care as much the shape, right? Like it just kind of has to match. So yeah, which model would explain enzyme with low substrate specificity? Induced, yeah, that it's more flexible, the substrate just kind of has to look like the active site to fit in, yeah. And so the lock and key model, like it works, but it doesn't explain things like that. With the lock and key model, it's the idea of like, oh, only one type of substrate will fit into the pocket. But what we observe is that enzymatic activity can be a lot more generalized. And I'll bring back the idea of what you did in that the carbohydrate lab to kind of bring together these topics for you. All right, so there we go. So my answer here, low substrate specificity, which means like kind of looks like substrate, kind of looks like active site. So that means a flexible model. So the induced fit would explain that. But if we had an enzyme that only works with, let's say, one type of sugar, like it only works with D-glucose, L-glucose wouldn't fit, that would be better explained by a lock and key model, right? That there's high substrate specificity. All right, so so that's how enzymes and substrates fit together so we can make the products that we want. Let's look into the details of how enzymes achieve that. Like, why do we need enzymes to speed up reactions in our body? So yeah, what makes enzymes good reaction catalysts? So remember, a catalyst just speeds up your rate of reaction. So speed up rate of reaction. That's from chapter 4. But we said it generically, like a catalyst will raise the reaction rate because it lowers the activation energy. So we'll look at the details of how catalysts lower the activation energy so it speeds up the reaction. So one of the factors, this is the whole idea here. Okay, so if I look at this graph right here, if we have some type of chemical reaction we want to do, like cleave a amino acid off of a peptide, sure. It takes energy to do that, right? It takes energy to break a bond. And so in the absence of a enzyme, like if we just wanted to just think about the energetics to break that peptide bond, follow the red line, all right? That's the activation energy for that chemical reaction without an enzyme. That's the energy just to, break that chemical bond and form the new bonds in the product. In the presence of a catalyst, we lower this first, hill that we have to climb. We lower it, so we make it easier for our bodies to do that energetically. And so in the presence of an enzyme, we just lower this top peak, that hill that we have to get across. But note here, see how where we end up in the same place? Right, the end result of an enzyme catalyzed reaction is we still have those same products that we meant to make. We just make it easier on us to achieve that. So one of the factors that allows us to lower the activation energy and push those reactions rates forward is proximity. Right, the active site, right, is where the chemical reaction occurs. So if you, like in our example previously with glucose and phosphate, if you're able to bring together two reactants that you need to react with one another, if you just think about it, if you have the two reactants next to one another in an active site versus spinning around, loose in a solution, it's much easier for that chemical reaction to occur if they're close to one another. And an enzyme helps us do that. Right? So yeah, here what you say, in the absence of an enzyme, we just have the substrates floating around. Who knows if they're going to knock into one another to make that chemical reaction occur. Versus in the presence of an enzyme, the reactants are close together, so they can exchange their atoms and electrons to make those new chemical bonds. Cool? Okay, so that's one reason why enzymes, but they're synonymous, are really good at making reactions go fast. They bring the reactants together. Other factors, orientation. So if you recall from chapter 4 when we talked about, or 3, I don't remember, we talked about chemical reactions. The reason there's an activation energy for a lot of chemical reactions is because we need to knock two molecules together, but they have to be in the right orientation. Do you remember that? Like if we bring two molecules together, not every collision will result in a chemical reaction. They have to come together, but in the right orientation in order for the products to be made. And so what an enzyme does is, again, the thought of proximity. When you have the two substances in the active site, they're close together, but because of the intermolecular forces that are holding those substrates in the active site, typically they're also in the right orientation for them to bump uglies and make your product. All right, and that's the whole idea of intermolecular forces, that, intermolecular forces hold your protein together in the right shape, but those intermolecular forces also bring your substrates to the active site and in a specific orientation. So that's what you kind of see right here. See these little, again, picture representation, that there's certain functional groups on the outside or in the active site of the enzyme, and they orient or they interact with the substrates in a certain way. So, it kind of splays out the sugar molecules so that phosphate group can come in and attack it at the correct orientation. And again, that lowers the activation energy here. And then, so that's orientation. That's why activation energy is lower. Also bond energy. So the idea of forming your enzyme substrate complex, so that's what you see right here. Once those intermolecular forces hold in your substrates, what happens is that it weakens the bonds in the substrate themselves. And so again, weakening the bonds because you're putting that substrate in the active site will make bond breaking easier for the next step in the chemical reaction. So here's the enzyme's active site, and so what you see right here, Mg2+. Do you remember, is that an enzyme? Is magnesium an enzyme? No, it is a helper. What kind of helper is it? Cofactor, yeah, because it's inorganic. And so what you see here, the purpose and how Mg2 plus is helping this chemical reaction occur is you can see Mg2 plus is in the active site with the enzyme and see how it's interacting with the phosphate group here. All right, and that's how it's helping. So what this enzyme is doing is that it's interacting with the sugar molecule, weakening the bonds. Magnesium here is weakening the bonds between these phosphate groups, so a new bond can be formed here, right, and make the product that you want. So that's what the purpose of magnesium is here, and that's why you gotta take your Flintstones multivitamins, all right, so your enzymes can do this in your body. So both of these things are reasons why enzymes are good catalysts. All three reasons are reasons for lowering the activation energy of the initial chemical reaction. That's basically it. So all three of those reasons. Okay, so keep going. All right, so let's look at some details of enzyme activity, meaning, you know, like what is the rate of products being formed when you introduce an enzyme to some type of chemical reaction. So here, this is the idea. We're going back to the ideas. If we have the enzyme and substrate in solution, chemical equilibrium can occur, and we have this enzyme substrate complex, and then at a certain rate, products are formed. So with this idea, what scientists do to kind of study enzyme activity is by changing the concentrations of the substrate. So how much, you know, reactant are you starting with? How much enzyme are you using up? Or sorry, you don't use up enzymes because you get it back. But if we vary the amounts of both, how does that change the amount of product that we make? So I think the book gives like a really good analogy here. So here's the analogy. If you have a bunch of bike parts, okay, like they're all broken or whatever, that's like your substrate, all right, your parts, that's your substrate. All right, and you have a bunch of workers who their goal is to put together those parts and make more bikes, like functional bikes, all right? So those workers are essentially how should I say this? The reaction without a catalyst, if you just have workers and nothing else. So the idea here is if you add tools and give tools to the workers here, then you essentially catalyze the reaction. That you give them tools, now they have the tools and knowledge to put together those parts and make bikes. All right, so the idea is having workers and workers and tools. We catalyze the reaction to turn bike parts into bikes. Because if I just had a pile of bike parts right here and no workers and no tools, so I just have a pile of bike parts, will that pile of bike parts spontaneously become bikes? No. We have to give the reactant something to help push the reaction forward. So that's the purpose of the workers and the bikes. Okay, so note here that there are 3 workers. All right, each one of those workers can make a bike in a certain amount of time, right? So those 3 workers have a certain type of output for bikes. I don't know, like a bike an hour or something per each of them or something like that. So the idea here is, okay, what if all these workers, these 3 workers are working on the pile of bikes and they have this certain output. They're like the enzyme, you know, chugging out finished products. What if I made the pile of parts larger? If I gave them more parts, would that make the reaction go faster? No. No. So the idea is that the workers and the tools and the bikes, with all of those together, you get a saturated concentration of enzyme substrate complex. And at that certain point where you're saturated there, adding more substrate won't change the output, the enzyme activity that you get. Because there's only, if you have a set amount of enzyme, there's only a certain rate that you'll get out. Just adding more substrate won't do anything. So that's the idea of steady state conditions, all right? Where you have like a set amount of enzymes, you have a certain concentration of substrate, of your reactant, but at a certain point when the pile is really, really big, your workers can only work as fast, and it has no effect on the output of bikes. So if we had 3 enzymes here, 3 workers, but only like a small pile of bikes, once they're done, right? So that's the idea of like, okay, at the initial adding more substrate, the activity goes up, but at a certain point you kind of get saturated, the pile is so big it piles up, and you just only have a certain rate going out. So that's the idea there. And this will help us when looking at inhibition, because there's different ways we can affect the activity or the rate of enzyme activity going forward. So yeah, under steady state conditions, when we've saturated the system with substrate with reactant concentration, the rate, your product is being produced as efficiently as possible, but the rate doesn't change if you add more reactant in those systems. So that's a good analogy there. All right, so with that in mind, keep the idea of the workers and their specific rate in mind, because there's ways that we can affect enzyme activity or the amount of product that are produced by different factors, and you've already seen some of them. So factors that affect enzyme activity. So remember, enzymes are proteins, right? Proteins are made up of amino acids, right? Amino acids have different functional groups, right? The state or the charge of the functional group is dependent on the, do we remember? Zwitter ion. The idea of the Zwitter ion, depending if the functional groups that are present on your amino acids, if you change the pH of the solution, sometimes you protonate your carboxylic acid, sometimes you protonate your amine. And so what happens there is that you change the structure, you change the primary structure of your protein, and therefore you will change your secondary and tertiary structure of your protein. So what I'm trying to get at is that pH, if you add more acid or add more base to your solution, is that you change what your protein looks like, and then it won't function anymore. All right, so enzymes only function at certain pHs, and you saw this in that car lab, and I want to talk through it, but I don't exactly quite know what you did because I don't know what results you got, so we'll have to just work through this together. But the idea here is that enzymes, they are most active at optimum pHs. This pH doesn't necessarily mean physiological pH. What is that? Physiological pH is like 7 point something, all right? So physiological pH is like 7.4, I think, or something like that. Not all enzymes operate at 7.4. A lot of them do. So for example, pepsin, we talked about the protease enzyme pepsin in our stomachs. Do you think pepsin works optimally at 7.4 in our stomach? No, our stomach is really acidic. So pepsin probably works most optimally at acidic pHs, all right? So if you change the optimal pH, you're going to change the activity of your enzyme. And it all has to do with chemical equilibrium, right? So this last chapter, chapter 9, and the beginning of this chapter, amino acids change their charge based on the pH that they're in. So what you're doing when you change pH is you're disrupting the functional groups that are on the ends of your amino acids, and therefore you will change the structure of your enzyme. All right, so basically just know, change the pH, you screw up the enzyme. So let's think about the car lab you did. So you were given sugar, disaccharides, right? Lactose and maltose, do you remember? No? Disaccharides, yes? Okay. And you introduced some enzymes to them, right? Lactase. We know lactase is an enzyme, why? It ends in A. So lactase breaks down lactose. Lactose is a disaccharide, right? It's made up of galactose and glucose. So what happened here is that, like, you had some lactose, the sugar, the milk sugar, you added lactase, and you tested the resulting solution for what? Do you remember what you tested it for? Glucose, right? So you think, if this reaction occurs, lactose, you add lactase, it breaks it down, and you make glucose, you would imagine that the concentration of glucose would go up, right? If this reaction occurs. Do you remember that? Did that happen? When you had lactose, you tested it for glucose, there wasn't a lot. but then you added lactose, or sorry, you started with lactose, you tested it for glucose, you saw very little glucose. Then you added lactase, the enzyme worked on breaking that lactose down, you tested the solution again, and you saw that the glucose concentration went up. Do you recall that? Yes, all right. So that makes sense. But then you did it again for maltose. Do you remember what maltose is made out of? I thought it was just 2 glucoses. Yeah, it was just 2 glucoses. Glue and glue. Okay. So you would imagine if you test maltose for glucose, you should read nothing, right? Because it's maltose, it's not glucose, it's maltose. So then you add an enzyme, in this case we added lactose again, or sorry, lactase. We started with maltose, we added lactase, then you tested the resulting solution. What did you see? Did you see the glucose concentration go up or down? I only saw one person's data, and I saw that the glucose concentration actually went up. Did it go up? Do you recall if it went up? It did not go up? You don't remember. I'm pretty sure it went up. But no, I have a point here, okay? We saw that the glucose concentration, did it go up? It went up, thank you, okay. Glucose, it went up. That doesn't make sense, but lactase only works on lactose. What is that related to that we just talked about? Lactose and maltose are both sugars, disaccharides. Don't they kind of look like one another? Yes. So what does that tell us about lactase? It's an enzyme that breaks down lactose, but it also works on maltose too. So what does that say about the enzyme specificity of lactase? It's low, right? That it can recognize both lactose and maltose to break it down. Does that make sense? Yes. So at first, maybe, when you're doing this lab, you're confused, like, why does this enzyme work on both of these things? And the reason is because of substrate specificity. Both of those sugars look like they're both disaccharides, so they both work, okay? Yes. So this example, the idea that both, when we add the enzyme to either sugar, glucose goes up, is the idea that lactase has a low substrate specificity, okay? What else did we do in that lab? We added acid or base, right? We did the same thing again, but we added acid or base. So when we add acid or base, what are we adjusting? The pH. So what happened when we did these same reactions, but we added acid? Did we see the glucose concentration go up? Think about what I just said. If we disrupt the pH, enzymes don't work. So if we adjust the pH, if we make it more acidic, we won't see the glucose concentration go up because we essentially disrupt the enzyme entirely and it doesn't work anymore. Do we see that? I'm sorry, that was so long ago. I'm sorry. We were talking about sugars back then when you worked on it, but now bringing it back because of enzymes. So yeah, the idea is when we added HCl or sodium hydroxide, so both of those conditions, we're either making it acidic or basic, we're changing it from its typical functioning optimal pH, we will see no increase, so no change in glucose concentration at all when we have the disaccharide and the enzyme together. All right, do we see that all come together? You see it all come together, yeah? All right. All right. Can I move on? All right. Does that clear up that lab? All right. Okay. So pH. If we mess up pH, the enzyme doesn't work. So we can just imagine if we mess up the temperature away from the optimal temperature of an enzyme, the enzyme won't work. Okay. So our bodies are typically the temperature of 37 degrees Celsius. Right? So most enzymes operate in that range. Enzymes that are in our body, okay, operate in that range. And so if we deviate from that temperature, you know, if we have a fever and our body heats up, enzymes won't work as well. If we get hypothermia, if we're really cold, enzymes won't work as well, and you'll probably get really sick. So yeah, at high temperatures, kind of the reason that enzymes don't work as well anymore, especially the high temperature ones, is because raising the temperature denatures proteins. We already talked about that previously. So if we denature a protein or an enzyme, will it work anymore? No, because its structure really matters, especially about that active site, in order for it to push chemical reactions forward. All right, so an example, real life example, as you know, I like to cook and stuff like that. When we think about yeast, all right, yeast contains enzymes that help make our bread nice and fluffy, all right? So it contains enzymes. So think of yeast, it contains, like the packets of yeast you get, contain enzymes. So when we want to, let's say, if any of you cook or use yeast, how do you activate yeast? No one makes bread? You put it in warm water. Yes, that's the big idea. And a little bit of sugar, right? Like a little bit of sugar to feed the yeast. But you add it to warm water, all right? Because raising the temperature of the yeast brings it to a more optimal temperature, so we can activate the yeast so it can do its job that we want. All right, so now I put you on the spot right here. If you have a big jar of yeast and you use only a little bit, where do you store the rest of that yeast? Use you shouldn't, if you want to waste your money, you put it back in the pantry. But typically, after you've opened a container of yeast, or if you're cheap like me and like use half the bag and you want to like save that half bag, even though a bag is like 50 cents, okay? If you want to save that half bag, I'll put like a paper clip over it and I put it in the, anyone want to think where to put it? In the fridge. Yeah, or in the dark too, that works too. Yeah, you put it in the, and whenever you want to save anything, you put it in the fridge, right? Same thing with yeast, because when we put it in the fridge, what we do is we cool it down, okay? We cool it down and we reduce the enzyme activity of the yeast so it doesn't kill itself. Yeah, so that whole idea that you cool stuff down, even the reason we put anything in the fridge, right, is to reduce enzyme activity so our food doesn't spoil, okay? All right, that's why we put stuff in the fridge. All right, yeah, so I did that, right? How do you activate yeast? You warm it up, put it in warm water. Where do you store an open jar of yeast? You put it in the fridge. Yeah. Okay, cool? Yeah, so temperature messes that up. Or if you want to bring this back to like our worker analogy, you know, if you have your workers, they're making the bikes, and then you decide to put them in like a hot yoga room, all right, where you make it like super humid and like 120 degrees Fahrenheit. Do you think they'll work faster in a hot environment or like, you know, just in like a normal room? No, if you put them in hot yoga and make them make bikes, they're probably going to slow down, okay? So it's the whole idea. There's like an optimum temperature, and then if you deviate from that, you're going to slow down your enzyme activity at the end there. Okay. Oh, but for those of you who do cook, okay, I said previously when you activate yeast, you put it into warm water, but do you put it into boiling water? Do you think that you like add boiling water to it? No, because that'll straight up kill it, all right? Because we want to warm up the yeast to increase its activity, but we don't want to warm it up so much that we like completely denature the enzymes. Yeah, okay, so there we go. Make bread, go home and make bread. All right, okay, all right, last ideas here. So other factors that affect enzyme activity. So this is how we kill enzymes, kill enzyme activities. And this is the idea of inhibitors. There's different type of inhibitors that we're gonna talk about, 3 different kinds here that all reduce enzyme activity. The first one is competitive inhibitors. So the idea is if there's other stuff in solution that looks like the substrate that we want to like change and become a product. If we have something else in there that looks like your substrate and can fit in the pocket of your enzyme, that is a competitive inhibitor, because what happens is when that other thing is in the pocket, your substrate can't come in, all right? There's already someone in there. So if you have a competitive inhibitor, they're taking up spots where the chemical reaction can no longer go forward with your substrate. So the idea here with competitive inhibitors is that they, one, look like the substrate. They look like the substrate, and they take up the active site. So the substrate can't go on there and do the chemical reaction. So what you'll see when there's competitive inhibitor present is that your enzyme activity will go down when there's a competitive inhibitor. But the thing is with competitive inhibition is we can reverse it by just adding more substrate. If we add more of the reactant we want, we essentially dilute the competitive inhibitor, the thing that's taking up the active site. We just add more of the stuff we want, and we should be able to see the activity restored. Because we're essentially just diluting that, that inhibitor that takes up that active site. All right, so that's competitive inhibition. We can reverse it, cannot interact with substrate and form product. Oh, I didn't write that, what I just said. So to reverse it, if we add more substrate, activity will return. All right? Because once we have more substrate floating around, there's so much, it'll just kick out those inhibitors that are taking up your active site. Because remember, making your enzyme substrate complex is an equilibrium. All right. So if you add more substrate, you'll push it more in making your enzyme substrate complex there. All right. So that's a competitive inhibitor. What you should know is that it looks like the substrate. And typically, you'll see this with things with low substrate specificity. So that enzyme won't care whether it has the substrate or the inhibitor on there. But only the substrate will make the product. All right, so if the inhibitor is there, it won't make product. All right, the next one is non-competitive inhibitors. So this has nothing to do with the active site. An non-competitive inhibitor is something that looks nothing like the substrate, but instead interacts with the enzyme. So it changes the shape of the enzyme. So if you change the shape of the enzyme, you change the shape of the active site, and the substrate can't go in and your reaction is not catalyzed. All right, so essentially I like to think of non-competitive inhibitors like this example is it spoils your enzyme. It changes the shape of it. All right, so the idea here is if you add more, so you see the inhibitor where it's going into like a different spot. So here is the active site where your substrate would typically go in. So instead, an inhibitor is just gonna like grab onto that enzyme, change the shape, and in turn changes the shape of the active site so the substrate can't go in anymore. So the idea here with non-competitive inhibitors, if we add more substrate, it's actually not going to change the activity of the enzyme because you've changed the shape of your active site. The only way to get rid of, to restore it would be to somehow get rid of those inhibitors, those non-competitive inhibitors that are attaching itself like a leech on your enzyme. All right, making it so it can't function anymore with your substrate. So in this example, if you add more substrate, it will not change. It will not restore your activity anymore. All right, so that is non-competitive. Okay, so the last one is irreversible inhibitors. So these ones sort of like the non-competitive in the sense that the inhibitor does not look like your substrate. In this example, your inhibitor comes in and attaches covalently to your active site so your substrate can't fit in. All right, so it's just literally blocking the active site, and typically it's covalently bound, and it will not let go. And it will essentially kill your enzyme. All right, so that's bad. Well, sometimes it's bad. I'll show you an example where irreversible inhibitors are good. All right, for humans. But yeah, so in this example, the inhibitor comes in, it actually does go into the active site, but it binds permanently. All right, so then that essentially just stops all enzyme activity within there. Okay, so there we go. those are the three types of enzyme activity, or the different types of inhibitors there. All right, so yeah, and here's the example. Sometimes you might want to inhibit enzyme activity. So the idea of antibiotics, it's like antibacteria, okay? So an antibiotic actually is a inhibitor. Specifically, it's a irreversible inhibitor. So what like penicillin is a compound that covalently permanently binds to the active site in an enzyme that forms these cell walls of bacteria. All right, so penicillin has a specific structure that it can go into the active site, binds permanently. And so in doing that, we deactivate the enzyme that produces the cell walls for bacteria, and so then your bacteria don't grow anymore. All right, and that's good for humans because we don't want bacteria, right? But it's bad news for bacteria because it kills them, but it's good news for us. All right, so that's the idea there. Bacteria bonds with penicillin, loses its catalytic behavior, and the growth of bacteria walls slow down, so that's a good thing. Okay. All right, so those are the factors for enzymes. Remember, enzymes are just proteins. Proteins are made up of small amino acid building blocks. All right, so let's start chapter 11. All right, so remember that this chapter 10, is due on Monday at midnight. They're short ones. These last ones are short, I think. I think they're like 45 minutes each. All right. Okay, so let's start chapter 11. And again, I took out a lot of parts of chapter 11. I wanted us to kind of focus on things that we already have talked about before, condensation reactions, hydrolysis reactions, in talking about the bigger picture here, so DNA and nucleic acids. So yeah, we're going to talk about nucleic acids here. So the idea of DNA, RNA, what is their role in our bodies? So why we need to talk about nucleic acids here is because this is how our bodies build proteins. So I told you before that, like, yeah, we make peptide bonds between amino acids. We make these long polypeptides. But who does it? Our bodies do it, and they need an instruction manual to do it using our DNA. So our DNA, which has nucleic acid in it, that DNA, RNA, they're made up of nucleic acids, contain the information for our bodies to build the proteins in the correct order. Do you remember how many amino acids there are? 22. 20 common ones, 22, yeah. So about 20 of them. So our DNA contains the code in order to tell our bodies, okay, we need to put those amino acids in this order in order to make like the keratin in our hair or whatever like that. Or, you know, the myoglobin that makes up our muscles. All right, it's completely different instruction manuals, and it's all contained in our DNA, and that is made of nucleic acids. So you might see a lot of reoccurring themes, okay, components of a nucleic acid. So remember, polysaccharides are made up of many monosaccharides. Polypeptides are made up of a bunch of amino acids. Nucleic acids are made up of a bunch of nucleotides all pieced together. All right. There's 4 or 5 different nucleic acids that make up our DNA and RNA. All right, so this is a little bit different, but still the same themes that we've seen in previous chapters. So so nucleic acid, that's like a strand of RNA, or, you know, a double helix of DNA. That's what we call a nucleic acid. So a nucleotide is one of the units in a nucleic acid strand. All right, so a nucleotide has 3 different components. It has a base, a nitrogenous base, a 5-membered ring sugar. We call it a pentose, pentose sugar ring, a pentose sugar, and a phosphate group. All right, so all 3 of these components are bonded together. By what type of bond do you think, or what type of reaction you think bonds all those? When we bring molecules together, what type of reaction happens? Condensation, yeah, so same thing. So this is a condensation reaction to bring these three pieces together, right? Okay, so these are the three components. So let's look at those different components. So the first one is of a nucleotide. So remember, nucleotide contains the base, the sugar, and the phosphate. So all three of those components. So we'll first look at the base component right here. All right, so the nitrogenous base, there's 5 different ones that we can have depending on whether we're looking at DNA or RNA, or we have these different bases. And so here they are, adenine, guanine, cytosine, thymine, uracil. Do those words sound familiar in bio? Okay, but in bio, did they show you what they look like? Oh, they did? No, maybe? So here in this class, we will inspect their intermolecular forces because there's reasons, like I hope if you took bio, you would know that like guanine pairs is the base pair with what other base? Cytosine, yeah, G and C go together. A goes with T in DNA. A goes with U in RNA. The reasoning though, like in bio, did they tell you why? No, they didn't, and it's because of intermolecular forces. So that's why this class is different, okay? Okay, so let me just show you the differences here. So we have different types of nitrogenous bases, like 2 families. We have the purines and the pyrimidines. All right, I like to remember this by purine is the smaller, like shorter word, but it has the bigger structure. Right? So that means the A and G, adenine and guanine have the larger rings because it has the shorter name. That's the type of molecule it is, versus the pyrimidines all have smaller rings. All right, and so that would be C, T, and U here. All right, so do know that. Do know the differences, which letters here are the purine rings versus the pyrimidine rings. So please do know that. All right, so because I do want to relate this to biology, because it seems like most of you know more biology than me, is that A goes with which one? T. Or U if we're talking about RNA, yeah, okay. G goes with C. But why? Why? Didn't you ever ask yourself why? Like in biology when you were told that, did you ever just think why? No? No? Okay, well, I'll show you why. So understand when we make nucleic acid strands in DNA, we have 2 strands, right, that come together to make your double helix for DNA. So what's happening is that, so for those of you who don't know biology, we will go into more detail here, but I want to make this relatable. Our adenine group and our thymine group come together and form a hydrogen bond, or at least one or more hydrogen bonds to kind of link those 2 nucleic acid strands together. So let's see where the spots for hydrogen bonding will occur. So on adenine here, so this hydrogen off of that nitrogen can hydrogen bond. Remember, this nitrogen also has a lone pair on it, so it can accept a proton and hydrogen bond with another group within there. So we said that adenine and guanine, they don't interact, right? They're not base pairs. A and G are not base pairs, correct? Right. Correct, they are not base pairs. And the reasoning, let's look at the spots that G can hydrogen bond. There are lone pairs on this oxygen. A hydrogen bond can form there. Here's a hydrogen off of that. There's a very electropositive hydrogen right there. And then this hydrogen off of that nitrogen can also hydrogen bond right there. See how there's 3 spots for hydrogen bonding on G, but there's only 2 on A. So let's look at the complementary bases. All right, cytosine. So G goes with which one? G goes with C. Cytosine, typically, yeah, we said goes with G. Look at all the spots for hydrogen bonding. How many are there? Do we see? There's 3. So see how the 3 on C and the 3 on G, they complement one another. All right, so oftentimes we won't see the 3 go with the 2 on adenine, and that's why adenine and cytosine aren't base pairs, right? And it's because of hydrogen bonding. All right, let's look at the next one. Thymine, right here. So typically the hydrogen bonding that occurs here with thymine are usually these two right here. All right, and so those are the two that we oftentimes see that create the hydrogen bonding. So that's why those base pairs are the way they are. They complement one another, and it's because of the locations of the hydrogen bonding on the base, the nitrogenous bases themselves. That's why A goes with T or U, and G goes with C. Okay, all right. So I went a little bit ahead there, all right? So, but from this slide, I do want you to know which one are purines and pyrimidines. That's the first component of a nucleic acid. All right, so first component, base. Next, we look at the sugar component, all right. So within the sugar component of a single nucleotide, there's two types that we can think about. RNA, do we know what that stands for? Ribose nucleic acid. DNA stands for deoxyribose nucleic acid. So that those names, the R versus the D in RNA and DNA, that's the sugar is what gives it its name. So if we look really closely at the ribose component, the sugar component of a nucleic acid, what I want you to focus on here is the group that dangles off of hydrogen number 2. All right, hydrogen number 2 of our pentose ring, so it's a 5-membered sugar ring here. So remember how we number carbons on a sugar? This is a little bit of a review, right, because it will be on the final. What do we call carbon number 1? Where does carbon number 1 start on? Do you remember? The rightmost carbon, which we call, has another name. Do you remember that name? The anomeric carbon, very good. So that's always carbon number 1. It's usually the one that's on the right. So we go 1, 2, 3, 4, 5. So I always like to think for these riboses and deoxyriboses that carbon number 5 is the tail on the ring, while carbon number 1 is the snout. It's the anomeric carbon on the tip on the right. Okay, so, but what I want you to focus on is what is the group that's dangling off carbon 2. So not carbon 1 or carbon 5. Look at carbon 2. Here. So off of carbon 2, we see for a ribose, the group that dangles off has an OH group. Yeah? But on deoxyribose, it only has H's. All right? So deoxy means that there's no OH group. And so what you need to focus on if you're looking at a full nucleic acid and need to know, is it ribose or deoxyribose? Is it in RNA or is it in DNA? You're looking at carbon number 2 on the pentose sugar. Right? So right here, yeah, so you see here, there's no oxygen that's bound to that carbon number 2. They're just both hydrogens. On this one, there is an OH group. So depending whether your nucleotide is made up of deoxyribose versus ribose will tell you whether it's RNA or DNA. Cool? All right. So there we go. All right, so the next part, actually we'll kind of just skip over it, is phosphate. There's a phosphate group on the end of it. So remember phosphate, we have this PO4 3- is the anion, but typically here we have phosphorus when we think about phosphate group in terms of organic compounds. We have this R group and oxygens surrounding it within there. So usually it looks like that. All right, so we have a phosphorus group with oxygen surrounding it. All right, so let's look a little bit more at the components. So combining them, so we looked at the sugar and the base. So typically what happens is we need to form a bond between the anomeric carbon on the sugar and on this portion of the ring on our base. So we see this is the base, and it's kind of making a bond with the carbon number 1 on our sugar. Just for fun, is this alpha or beta? Alpha or beta? This is like a little bit of a review. If we put the ring on this side, is that bond right there, is that alpha or beta? Yeah, it's beta because if we look at the tail on this side, you always look at the tail and look, is the thing that you're attaching, are they bumping uglies? That's unstable, so it's beta. Alpha is when they're opposite, okay? And because that's more stable. When they're opposite, so they're as far away as possible, so this would be beta. All right, that's just like a side thing. All right, so we form a bond right there through a condensation reaction. So what's highlighted here is the water that we're losing. Again, condensation reaction, we lose a water, we bring together two components. And so we form a glycosidic bond right there. Doesn't that sound familiar? All right. So we combine two things, condensation reaction, have one new molecule, and then that water. So now the next step here for forming a nucleic acid, we only have two of the three components. So this molecule right here is not a nucleic acid yet, or sorry, it's not a nucleotide yet. This is actually called a nucleoside because there is no phosphate on it yet. Phosphate. So right here, when we just have the free nitrogenous base, we call this adenine. But when we add our ribose sugar to it, we make it what we call a nucleoside because it doesn't have the phosphate group here. So then we call it adenosine instead because it has that sugar group attached to it because there's no phosphate. So I think on the next slide right here, naming nucleotides. When we have the base, the sugar, and the phosphate, we call it a nucleotide. But when it's just the base and the sugar, so like in this example up here, we call it a nucleoside. So that's just a small little detail. So again, no phosphate. Here. All right. And so here are all the, okay, I'll back up a little bit. Here are the different examples of our 5 different nitrogenous bases and the names that we would give them. Depending on, well, one, what type of sugar do we put on it? Because remember, within our sugars, there's 2 types. There's the ribose and the deoxyribose. So there's 2 different structures we can have there with the sugar, and then the phosphate group. So what's interesting here with the phosphate group, typically when we change the name, essentially we're just saying, okay, what nucleoside is there, and then how many phosphate groups are attached. So right here is our phosphate group. And we are attaching it to carbon number 5 off of the pentose sugar. And so we would call this nucleic acid adenosine monophosphate. So adenosine is naming this the nitrogenous base and the sugar, and then monophosphate is just telling us, okay, how many phosphate groups are on here. So if we would change this structure, so right here, note this ribose sugar is which one? Is it ribose or deoxyribose? Just regular ribose because it's OH. But if I change that to an H, this now is called deoxyadenosine monophosphate. All right, so right here, deoxy, that tells you what type of sugar it is. Adenosine tells you what type of nitrogenous base it is. And then monophosphate just tells you how many phosphate groups are attached there. So if this would be, and then we abbreviate it here, AMP to mean adenosine monophosphate versus DAMP, which where the D implies the deoxy. But if this was ADP, what do you think that would mean? Diphosphate? Yeah. So ADP, here we're saying it's monophosphate. So if we wanted to say diphosphate, so meaning if there was like another phosphorus group on the end, we would call it ADP. So that those 2 last letters just tell you how many phosphorus groups are on there. That's not something you would see as commonly. So here are all of them. So really what's changing with all of these different structures are which nitrogenous base are there. So a quick question here. Uridine monophosphate, do you think a DUMP exists? Uridine, no, because U goes with which type of DNA, like DNA or RNA? RNA. RNA stands for ribose nucleic acid. So UMP here, there would be no deoxy because the ribose that we have for U here is only ribose and not deoxyribose. All right, so DUMP would not work. Oh, I put that there. So UDP would mean diphosphate. That's all that means. All right. But yeah, the deoxy would not exist. And the same idea over here, if we look at thymidine here, so deoxythymidine monophosphate, so we would not see just a regular TMP because thymine does not go with ribose, it only goes with deoxyribose in DNA. So right here is a good place for us to stop. So everything I just said is on this list as well, how we put together the bases, the nucleoside, so that's without the phosphate, and then the nucleotides, that's with the phosphate, all right? And those are the different names.